amazing day, my dear STEM Legend learners! Welcome to our first episode for Quarter 3 here in Senior High Tech TV. I am glad you are here to join me in learning today. I am Teacher Ernest, and together, let us discover the exciting world of biology. If you are ready to learn, please behave, listen, and take note of the important concepts of our lesson for today.
It's trivia time!
Did you know that the first genetically modified animal was created in 1973? An enormous breakthrough in GMO technology came in 1973 when Herbert Boyer and Stanley Cohen worked together to engineer the first successful genetically engineered organism. It was a mouse. Are you excited for another amazing biological science adventure? Let's begin! Before we start, let's play a game entitled Choose the Best. I will show you a statement. Choose the best answer in 5 seconds. Are you ready? Let's go!
Number 1. A scientist cloned a goat. Which of the following statements is true about a cloned goat? Letter A. It has a new genes and traits. Letter B. It lacks the genes for reproduction. Letter C. It looks the same but different genes. And letter D. It has a genes that are identical to the original goat. Excellent! The correct answer is letter D. Number 2. DNA containing genes from a foreign donor is called blank. Letter A. Gene. Letter B. Plasmid. Letter C. Recombinant DNA. And letter D. Restriction enzymes. You are correct. The answer is letter C. Number 3. Who was the first adult mammal successfully cloned? Letter A. Agouti goat the mouse. Letter B. Copycat the cat. Letter C. Dolly the sheep. And letter D. Donald Trump the Donald. If your answer is C, then you are correct. Number 4. How do scientists cut DNA into smaller strands? Letter A. Agarose. Letter B. Ligase. Letter C. Really small scissors. And letter D. Restriction enzymes. You're right. The answer is letter D. And number 5. Any change or mistake in DNA replication can cause a blank. A. Cancer cell. B. Disease. Letter C. Mutation. And letter D. Virus. Precisely. The answer is letter C.
Let us discuss this further, so stay focused and don't go away.
Our lesson for today is all about genetic engineering. And for the learning competency, outline the processes involved in genetic engineering. At the end of the lesson, you are expected to define genetic engineering, explain the principle of recombinant DNA technology, and enumerate and explain the processes involved in genetic engineering.
Do you believe that organismal traits can be changed or altered? How can this be possible? Can traits be transferred to other organisms? If you are that curious, why don't you come and join me? Let's dive in the world where the naked eyes cannot see and be part of this invisible world that affects the visible environment.
The central dogma of molecular biology explains the flow of genetic information from genes to protein. It provides a molecular mechanism in understanding how genotype translates to phenotype. It became apparent then that changing an organismal trait is possible by altering its genetic makeup.
Let us define first the following terms. What is genetics?
Genetics is a field of biology that studies how traits are passed from parents to their offspring. It is the study of how living things receive common traits from previous generations. These traits are described by the genetic information carried by a molecule called DNA. The instructions for constructing and operating an organism are contained in the organism's DNA.
What is DNA?
Every living organism on Earth has DNA in its cells. Deoxyribonucleic acid or DNA is a molecule that contains the biological instructions that make each species unique. DNA, along with the instructions it contains, is passed from adult organisms to their offspring during reproduction.
What is genetic engineering?
Genetic engineering is artificially copying a piece of DNA from one organism and joining this copy of DNA into the DNA of another organism. It is defined as a direct manipulation of an organism's genes, including heritable and non-heritable recombinant DNA constructs. Genetic engineering involves the use of molecular techniques to modify the traits of a target organism. The modification of traits may involve: number 1, introduction of new traits into an organism. Number 2, enhancement of a present trait by increasing the expression of the desired gene. Or number 3, enhancement of a present trait by disrupting the inhibition of the desired gene's expression.
What is recombinant DNA technology?
Recombinant DNA technology is one technique wherein a gene of interest from one organism is inserted into a genome of another. This involves gene cloning using a bacterial plasmid as a vector. It is widely used in improving crop varieties. Recombinant DNA technologies developed in the latter half of the 20th century include the chemical splicing or recombination of different strands of DNA, generally using either bacteria such as E. coli or bacteriophages, or viruses that infect bacteria, or by direct microinjection.
And what is recombinant DNA?
The altered DNA is called recombinant DNA.
Here are the general outline on the formation of recombinant DNA. Number 1. Isolation of a specific gene from a donor.
The first step involves breaking open the cells of the donor to release the DNA and isolate the gene of interest. Letter A. Cells are broken open using chemicals and enzymes. Donor DNA is extracted. Letter B. Genetic probe is added. A DNA probe consists of a small fragment of DNA labeled with an enzyme, a radioactive tag, or a fluorescent dye tag. The probe will bind to a complementary DNA sequence by base pairing. Letter C. Reveal position of the gene of interest. This works this way. The DNA from the bacterial cell is released. A bacterial cell contains a circular loop of a DNA called a plasmid. The plasmid is isolated from the bacterial cell, and the plasmid will act as a vector for carrying a new gene. Example, the gene of the donor will be inserted into the plasmid DNA.
Number 2. Cutting or cleavage of DNA by restriction enzymes or RES.
It is put this way. The donor DNA and plasmid DNA are cut using enzymes called restriction enzymes. Restriction enzymes recognize a specific sequence of bases. It acts as a molecular scissor to cut the DNA strand within the recognition sequence. The donor DNA and plasmid DNA are cut using the same restriction enzymes. Restriction enzymes cut the DNA from the donor at specific points. The cut ends will have sticky ends. The same restriction enzymes cut the DNA from the plasmid, also revealing sticky ends. As a result of cutting by the restriction enzymes, the donor DNA and the plasmid are left with unpaired bases known as sticky ends.
Number 3. Ligation.
Ligation is rejoining cut fragments of DNA and forming artificial recombinant molecules, joining together of the gene of interest. Example, from animal with the vector, cut bacterial plasmid.
Number 4. Transformation.
Transformation is a transfer of the recombinant plasmid into a host cell that would carry out replication to make huge copies of the recombined plasmid, recombinant DNA introduced into bacterial cell.
And number 5. Expression.
Plasmid will produce the polypeptide coded for the donor DNA. Bacterial cell reproduces by binary fission. Bacterial cell produces the polypeptide coded for the donor DNA.
Now, we have already discovered the processes involved in genetic engineering. If you really understood our lesson, let us check your understanding by answering this multiple choice activity. Choose the correct answer from each question. Bring out your pen and squeeze notebook and let's begin. Are you ready?
Number 1. What branch of biology studies how traits are passed from parents to their offspring? Letter A. Botany. Letter B. Genetics. Letter C. Taxonomy. And letter D. Zoology. Very good. The correct answer is letter B. Number 2. A molecule that contains the biological instructions that make each species unique is called blank. Letter A. ATP. Letter B. DNA. Letter C, RNA. And letter D, RUBP. Very good. The correct answer is letter B. Number 3. What is used to cut donor DNA and plasmid DNA? Letter A, electrophoresis. Letter B, restriction enzymes. Letter C, restriction site. And letter D, splicing. If your answer is letter B, then you are correct. Number 4. The ability to combine the DNA of one organism with the DNA of another organism is called blank. Letter A, GEO. Letter B, GMO. Letter C, ligation and insertion. And letter D, recombinant DNA. You're right. The answer is letter D. And number 5. What stage in the formation of recombinant DNA is involved when cloning cells are opened using chemicals and enzymes? Donor DNA is extracted and genetic probe is added. Letter A, cutting. Letter B, expression. Letter C, insertion and ligation. And letter D, isolation. Exactly. The answer is letter A.
To sum up, genetic engineering is getting importance because of its use in different fields. In medicine, genetic engineering has been used to mass-produce insulin, human growth hormones, polystim or treating infertility, human albumin, monoclonal antibodies, anti-hemophilic factors, vaccines, and many drugs. Congratulations, my dear STEM Legend learners! I hope that you learned a lot today. Once again, I am your energetic teacher saying, eagerness to learn will lead you in the fulfillment of your dreams. See you in the next episode!